Hi and good morning. My name is Adam Carlson. I'm a equity research analyst here at ABG, and it's my pleasure to welcome you to our second Private Companies Pre-IPO seminar. We have 7 companies presenting today in half an hour slot, starting now and wrapping up at around 1:30. Each presentation will be followed by a brief Q&A, and all presentations will be made available to rewatch later as well. Our first presenter is Johan Living, CEO of XNK Therapeutics. XNK is a private Swedish cell therapy company sprung out of pioneering NK cell research here at the Karolinska Institute in Stockholm. They are in phase 2 clinical development with their lead NK cell therapy. I'll hand over the word to you, Johan.
Thank you very much, Adam, and a pleasure to be here. So here's our disclaimer. Please read it carefully. XNK Therapeutics is built around the belief that we can restore a part of your own immune system to its former glory, that we can make the natural killer cells cytotoxic again and start killing off cancers. As Adam says, we build around the world-leading research of NK cells that's been going on in the Karolinska Institute since the mid-70s. At our core, we have a platform technology that is both stable and versatile and allow us to develop therapeutic options. We have presented first in human phase 1/2 clinical data in multiple myeloma, in which we believe we have a good safety profile of the product and a promising efficacy. This promising efficacy made us go into a randomized phase 2 study where we combine our autologous NK cells with Sanofi's anti-CD38 antibody Sarclisa, and we're doing the study here at Huddinge. You might have seen that this morning we sent out a press release. We are expanding our platform technology to look into new cancers, and we are going to do that in collaboration with the MD Anderson Cancer Center, the world's most prominent cancer research. And we're going to target AML patients. And we're going to try to do a study where we can identify patient populations that can be targeted for a clinical trial with our autologous NK cells. And the belief is that these autologous NK cells will give a more persistent response. So we're working with natural killer cells. These are part of the body's primary immune system, and they work by recognizing patterns on the cell surface. So basically, they see a healthy cell and that pattern, and they say, this is a healthy cell, let's leave that. However, when a cell gets infected by a virus or transformed into a cancer cell, that pattern will change at the cell surface. The NK cells would recognize that and then kill that cell off. When you get cancer, you get a massive amount of these cancer cells into the body, and the NK cells, they stop functioning. They don't recognize these cells as being transformed into something else, and they stop killing them off. What we do, and our belief, is that we have the ability to restore the cytotoxic capacity of those NK cells and make them into a therapeutic option to treat cancer again. The team behind the company, we have a diverse board of advisors. We have the scientific part with the prominent researcher Hans Gustav Ljunggren, but we also have the entrepreneurial mind of David Berglund and the big pharma commercial guy of Steve Wooding. To our help, we also have advisors. The founder of the company, Evren Alici, is one of the top 10 researchers in the world in this field. And we also have clinical advisors that are going to help us with the clinical development. Ola Landgren is a prominent multiple myeloma researcher in the US now, writing international guidelines. And Fredrik Schjesvold, the current head of the Nordic Multiple Myeloma Study Group. We also have an experienced management that really gets things done. If you look at our technology and our platform technology, and step by step, the patient will come in and donate 1 unit of blood. That's 450 milliliters. That blood is then shipped to us when we freeze it. We can then plan the start of the production, the expansion and activation of NK cells. That's step 3 and step 4. When we're done with the production, we can freeze the end product, and we can store that up to 10 years in our freezers. When the patients will get the treatment, we ship it frozen to the clinic, and where we can infuse the cells bedside. That's a process that takes around 15 minutes. The patient will stand a couple of hours for observation and then go home. So from the patient perspective, everything is happening in an outpatient setting. Being an autologous therapy, we can also do multiple treatment. We can retreat the patient because we do not have to immunosuppress. Let's look at the platform technology itself. That's step 3 and step 4. It's a feeder cell-free system, which basically means we don't rely on any external cell lines. We only use the body's own cells. And we started from frozen material. These two things means that we can plan it and we can get the cost down quite significantly because we don't have to rely on other materials. Furthermore, from a regulatory point of view, it's probably an easier path because we don't gene modify these cells either, and we don't rely on any third-line cancer cell lines. If you look at the process, the picture to the right, and you have days at the x-axis and the number of cells, logarithmic scale on the y-axis, we start by optimizing the environment for the NK cells, and that takes a couple of days. After 5-7 days, the NK cells start to become cytotoxic, and after day 10, you see a rapid expansion of the number of NK cells. Then at day 20, we will have the number of cells we need for the therapeutic dosages, and we can harvest the cells, and we then put that into the freezer. The good thing that would happen over this period of time with the cells that they upregulate certain markers on the cell surface and downregulate others. One of the things that they downregulate is the CD38 expression, which basically means that if you combine this with the CD38 antibody, the natural killer cell will not be targeted by that antibody. So what happened in vivo then? Well, if we expose these activated NK cells towards your own multiple myeloma cells and you look at the graph to the left, you see that at day 0, that's the curve at the bottom, they don't kill off any cancer cells. After 5 days, that's the curve in the middle, they start to kill off some cancer cells, and they increase that capacity quite significantly at day 20. But these are natural killer cells. You want them to recognize the differences of a cancer cell and a healthy cell. You don't want them to kill off healthy cells. And that's what you see on the graph to the right. So if you take these activated NK cells and expose them to your own healthy cells, you see that at day 0, they leave them alone, day 5, they leave them alone, and at day 20, they still don't kill them off. So you have a very targeted killing towards these cells that has a change in the pattern on the surface. So we have patented this technology. We have 2 patents, one for the methods of expansion and activation, and one for the treatment of multiple myeloma. We have 2 patents in pending, one for a combination treatment with a high-dose antiviral treatment, and a new patent that we have submitted for our updated process. Over the years, we have also gained orphan drug designation, both in EU and in the US, which will give us market exclusivity at launch. Working with cell therapy, production is always a critical element. Today, we produce for the clinical trials at Cura in Huddinge. The trial is running at Huddinge, so it works well. However, to really facilitate the larger clinical trials, European, global, multi-center trials, we need to build up the GMP facility our own. We started that process in the summer, and we believe the actual construction will be ready after New Year's. When we have the production set up, we will be able to produce to around 100 batches per year, but we can easily scale that up if we need. This will also help us to understand the process and optimize it further for bigger production sites. Being a feeder-free system and also working without gene modification means that the process is relatively simple, so we can get it quite competitive in production cost. So where have we taken this technology then? Well, as I said, we started in multiple myeloma, blood cancer. In the summer, we started to look into different other indications, cancer indications, where it would make sense to have an autologous NK cell treatment and where the conditions are favorable. And as I said, as of this morning, we sent out a press release where we start to look into AML. But there are quite many of those cancers where you have a good fit for an autologous therapy, and the conditions for that technology to work is good. Specifically, we're looking for a relatively low tumor burden, a minimal residual disease setting, and also if you have an antibody using the body's own natural killer cells, called the ADCC mechanism, like Sarclisa, there's a good fit. We believe that there's going to be synergistic effects. So, looking into our clinical trial data, we have presented first-in-human phase 1/2 data last year at EHA, looking into safety and tolerability. And we treated 6 patients with 18 dosages in multiple myeloma. The primary endpoint was safety. We did not see any serious adverse events, which is pretty rare in the patient population like this. The majority of the adverse events were of grade 1, and the majority was unlikely to be related to our treatment. The most frequently reported AEs were shingles. The first 4 patients developed shingles. The good thing about shingles is it can be prevented and even treated. So we gave high-dose antiviral treatment to the last 2 patients, and they did not develop shingles. The other frequently reported AEs were upper respiratory infection, lumbago, diarrhea, headache, and paresthesia. We have a biomarker called granzyme B. When the NK cells are killing off their target cells, they release granzymes. The half-life of granzyme B is around 20 minutes, so if you can detect it, it's a marker of a killing process going on. And we believe the NK cell then is killing off the target cells. So the graph to the left, you see the levels of granzyme B in serum, and it goes up when you get the dose, and then it declines. And then when we get the second infusion, it goes up and goes down again. To us, that tells us that the NK cells are coming into the bloodstream and then migrating into the tissue. That's exactly what we like to see. To the graph to the right, at bone marrow aspirates, we have the levels at diagnosis and then 3 weeks after the 3rd infusion. And we can see increased levels of granzyme B in the bone marrow. To us, it tells us that the NK cells have migrated into the bone marrow, where you have the multiple myeloma cancer cells, and there is some killing going on. So what is then the effect of patients? Well, there is a measure for clinical response, basically of the tumor burden. It's called M protein or M component. And where we can measure it, we see a decreased level of that in all the patients. When we look at the bone marrow, we also see a decreased level in the bone marrow. So for us, we see a sign of a clinical response. What matters to the patient is obviously how long time it will take until the cancer will come back. That's progression-free survival. But then also overall survival. How long do I live? And we have reached a median progression-free survival, and that is of 34 months. Good to know 2 patients still haven't progressed after a median follow-up of 60 months. After 60 months, median follow-up, all patients are still alive. To us then, this was all arrows of the efficacy were pointing into the right direction. We believed this was a safe drug, and we took the decision to go into a phase 2. So this summer, we launched a phase 2 trial, a randomized phase 2 trial, where we combine our autologous NK cells with an anti-CC antibody called Sarclisa, which is an anti-CD38 monoclonal antibody, utilizing these ADCC pathway, the natural killer cells in the body to do the killing. So by adding a large amount of highly activated NK cells, we believe in synergistic effects. 60 patients should be recruited, and the primary endpoint is overall response rate, including a minimal residual disease. A large variety of secondary endpoints and exploratory endpoints. Patients are currently being recruited, and to the right you see some pictures with our staff actually producing the drug for the patients in the trial. Today, 4 patients have been recruited. It was a bit slow in the start, but now the recruitment pace is picking up nicely. During this summer, we also said we're going to expand and look into other indications where it would make sense to have an autologous NK cell therapy. And as of this morning, we sent out a press release with an agreement with MD Anderson Cancer Center regarding AML, or leukemia. MD Anderson is the world's most prominent cancer center, and together we are going to find the right patient population for an autologous NK cell therapy. And when these criteria has been established, we're going to try to take that into the clinic. We believe that an autologous NK cell therapy in the field of AML makes sense because you can get a more persistent response. Your own immune system will not clear out these NK cells. They will be there and killing off these AML cancer cells. So, just to sum it up, XNK Therapeutics, we fundamentally believe in your own immune system, in the NK cells, and our ability to restore that as a therapeutic option. We are built around the world-leading research of NK cells that's been going on at the Karolinska Institute since the mid-70s. At our core, we have a platform technology that allow us to develop therapeutic options for different cancer indications. We come quite far. In multiple myeloma, we are in phase 2, a randomized phase 2, in collaboration with Sanofi and Karolinska Institute and Hospital. And today, we announced a further expansion of this platform into AML. So thank you very much. Questions?
Great. Thank you very much, Johan, for that presentation. A few questions, if I could. Maybe a first one, just kind of on the basics of XNK. So you're using NK cells from each individual patient's autologous as opposed to kind of from a universal donor allogeneic NK cells. And just to kind of understand the kind of advantages or potential disadvantages, many people will be familiar with kind of with the benefit of not needing a immunosuppressive conditioning treatment. Maybe explain that a little bit, and if there are any other kind of obvious advantages or indeed disadvantages to either approach.
Yeah, that's a very good question. Thank you. So being an autologous therapy, the good thing, as you said, you don't need to immunosuppress the patient because you just put back your own cells. That also means that you can give repetitive dosages. So you can maintain and retreat as long as you need. Another potential of being autologous is what we're trying to explore in AML here. Potentially, you have a more persistent response because your own immune system would hopefully not be a threat to these cells. They will just flourish within this environment. Another good thing is that this is NK cell biology. You have an optimal licensing of these cells. They will learn to recognize your own cells optimally because they are your own cells. So that's kind of obvious. Another thing that is on the upside of using autologous is that, at least I believe, that the regulatory pathway will be much easier because you don't transplant, you don't use another donor. So I think it's an easier path. That's my personal perspective. Being autologous, using the cells from another individual, of course, has the advantage of you can produce in advance. So, I mean, it will be there where you need it. And there are certain conditions where that is absolutely needed. You have an aggressive cancer you need to treat now. But in other cancers, and depending on where you are, well, that need is not obvious. So you can actually wait, and then an autologous could make sense.
Okay, great, thank you. And maybe staying on that point of kind of speed of production and availability and so on, at least initially, the first generation of cell therapy products were kind of hampered by the logistical challenges of creating the cells and shipping them and all of that. It's been a few years now since those therapies were launched. How much has the field progressed, and how much kind of better placed is the healthcare system as a whole to kind of handle the new challenges of a cell therapy?
Yeah, CAR T are in autologous treatment as well. And when you launched that first, the hospital was not ready. It was, you need to basically train the hospitals to understand how to deal with it. But they've done the job now. So I think today, the bigger hospital, they are utilizing CAR Ts and they're quite familiar with the processes. We have a much simpler process. Basically, you take a bag of blood and then ship it to us. And today we have 24 hours until it has to arrive to us. We're trying to expand that further. So that is one thing. But at the end, for our process, we can freeze the starting material. That means we can schedule production. So we'll have everything within our control. And then, of course, when we ship it back, it's also frozen. And we have 10 years of stability, so we can basically store it for a long time if needed.
Okay, no, interesting. And there's obviously been a lot of development within the NK cell field, NK cell therapies in particular. And I guess the two axes that companies are differing on is the autologous versus allogeneic, and some are also pursuing kind of a gene editing approach, trying to add some kind of beneficial features to it, and others aren't. And it seems like there are potentially pros and cons with those approaches. I'd be interested to hear kind of what the benefits and potential cons would be of being a non-gene edited NK cell therapy. Yeah,
so we are one of the leading companies within the non-gene editing field. Of course, you can gene edit. We could do that as well. We haven't seen the need for it yet. You could go CAR NK, but we are utilizing these ADCs and component antibodies. Basically, you get the same thing. So we're utilizing other other drugs, other antibodies to make the work for us, basically. That's where we're going today. Where we go in the future, I don't know. We could also go down the allo path because, I mean, it's just giving my NK cells to someone else, right? They have an allo treatment. So, but we believe that autologous NK cells is a very good place to be.
Okay, good. And on the point of kind of the burgeoning NK cell field, there's obviously been a lot of interest from big pharma as well. A lot of the development and innovation is coming from kind of smaller biotechs such as yourselves, but big pharma have been kind of quick to make collaborations and acquisitions and so on. We've seen well, we've got Johnson & Johnson with Fate, we've got Sanofi with yourself and Kiedis, and yeah, the list goes on. Amgen with Nkarta and so on. But you've obviously got this phase 2 collaboration with Sanofi now. How are you thinking about kind of partnering and further development, I guess, both in the near term, but also long term?
Yeah, well, we just launched a collaboration with an academic center today. We fundamentally believe that moving forward and selecting indications, if we can find a partner, of course, that's a much more stronger proposition than going alone. So we will talk to other, quite, on a frequent basis, trying to understand if they see a need for an autologous treatment and if they have something they can offer us, right? So it have to be a win-win. We started this work in the summer, and I'm pretty proud of the way it has progressed so far.
Good. And maybe just a question on the announcement this morning. A preclinical collaboration at this stage anyway with the MD Anderson Center on AML. Perhaps a question on kind of how that came about and what kind of is the incentive from your side, but also from the MD Anderson side. What are they presumably excited about in terms of what you guys are developing and what kind of drove them to enter into this collaboration?
Yeah, no, for us, of course, it's exciting to develop our therapy outside of multiple myeloma, and AML, or blood cancer, is a close proximity to what we're doing. What the team at MD Anderson really would like to see is a persistent response. They believe that they can get that from an autologous therapy. So that's their drive, the patient benefit, and that drives us as well, because AML is not a nice cancer to get. I mean, the 5-year survival is what, 25%. It's a devastating cancer, and if we can do something in there, and it would make sense for us to develop our therapy, which we believe it would, well, it's a good place to go after.
And you mentioned the kind of the persistence of the cells in your presentation, I think, as well, and how that could kind of feed into the durability of the effect that you would get with an autologous as opposed to an allogeneic cell therapy. And I guess there's a theoretical rationale behind that. Is there, what have we seen in terms of clinical data so far that would suggest that sustained effect may be an issue with allogeneic? Has there been much data on that so far?
Coming out some data, clinical data, where the belief was that you have a shorter persistence that you would expect. So there are some, but they are very, very early on. They came out some clinical data, phase 1, from an autologous company in lung cancer, I think it was a month ago, where they combined with another antibody in lung cancer, and they show a PFS of 9 months compared to the antibody alone of 2 months. So a dramatic effect. So the combination of an autologous therapy with an ADCC-competent antibody seems to be a very, very, very good fit, and that's what we're trying to look into now in multiple myeloma. And we'll probably find other sweet spots for that as well.
Good. And maybe just a final question. You're obviously an unlisted company. What can you say about kind of your cash position, funding needs, and plans going forward?
Yeah, well, we took in 64 million Swedish krona in, I think it was in April. So we have sufficient funding right now. Where we're going to go in the future, of course, no one knows. It depends on the market climate and what happened with the company. But what I can say is I'm very proud of the pace we currently have within the company, the deliveries that we've done. The management team, as I said, we are a great team, and we can get things moving.
Okay. In the interest of time, I think we have to wrap up there. Thank you very much for an interesting presentation and Q&A, Johan.
Thank you very much.
Okay, good. And I'll pass us back and over to the next presentation in just a few minutes.